So, as I said in the previous slide or previous lecture, we will we have understand only enzyme catalysis, how it's the two theory involves and induced fit and Emil Fisher's proposed lock and key model. Okay. Similarly, what is the specificity is that the question can be asked, write down a theory of enzyme catalysis, then write a specificity of enzymes and their mechanism underlining editing. Then it can also ask, write a short note on concept of active sites and in detail description of enzyme substrate complex. Sometimes they are also asking, write active site mapping. What do you know about active site mapping? Explain in one words with give definition also. Factor affecting catalytic efficiency that can also be asked in the examinations. And MIchaelis menton equation means mechanism of enzyme reactions, how zero order reaction is happening. Then detection of intermediates in the enzymatic reactions. So one by one, before understanding this, I will give you a certain reaction of different order, or you can say kinetic summary of any enzymatic reactions. So here you will find features. It include reaction types, rate of expressions. You can see the direct plot of the rate with a or integrated rate equation, linear plot equations with that graph, then half-life expressions, then plot also, and unit in meter per second or unit of K MCAS systems is given in that. For example, in zero order reaction, if you see the reaction type A or substrate finally or reaction of the order, it will give us to product. Same thing is in first zero order and first order, same it will convert into a product. But if you see the second order reaction, it require A plus A. So you can write like this. If you see the rate of expression, that K into bracket A degree, okay, zero order reaction, therefore A degree or A zero. Similarly, rate if you write in the first order reactions, you have to write A1. Similarly, if in second order reaction, the rate will be A2 or A square. Now, now you can see here direct plot. From rate A, you will find finally in the linear reactions with zero order and it will give you a product. If you see in first order, that will show you like a graph, increasing rate. So that is K into A1 or inverse 1. Whereas in case of same graph, you will see with rate versus A. Now, if you integrate the rate of reaction for the zero order, first order, and second order, that will ultimately change a log. In first, you will zero order, there is no logarithmic changes will observed, whereas in case of log A and here one half A with that reaction of K minus K divided by 2.303 and here is also K. So these reaction you have to remember not by words but by photographic memory. You have to remember the linear plot and direct plot. The reaction you will find in the direct plot in between one square you have to write zig. In between this one at 90 degree, 90 by 45 degree, both 45 degrees. Here you have to change with the log A0 or log A0 and 1 half 1 upon A, like its reverse one. Similarly, T 1 half plot, you have to show a different reactions. These first order will come here, T half, same reaction. Similarly, in the direct plot also, that only the name is changed, T half A and 1 half A, and unit you know very well. So you can write rate of different order a kinetic summary. If it is asked in the examination, you can draw only without any explanation. Write differences between zero order, first order, and second order. Or they can also write zero order or second order. So you can write like this. Now we will start primary things. As I say, what is that? Tell me which point.
Mechanism, specificity and elasticity mechanism.
Mechanism by which enzyme catalysis reaction. So, specificity of enzyme involved can vary depending upon the type of reaction and specificity of enzyme involved. So, however, there are some common mechanism that enzyme employ to facilitate the catalysis. One such mechanism is the formation of enzyme substrate complex, where the substrate molecules bind to the enzyme at active site. So you can see in this scarlet color enzyme is given. Here is the site, active site, plus S, this kidney like structure substrate. It will bind like this enzyme substrate complex. And finally what will happen, wherever you will find the active site, that color is cut into that product and ES complex. So similarly in these blue reaction, see here, you can with the hexagon, you can understand what is, for example, substrate you have a sucrose. Sucrose is made up of, you know very well, glucose plus fructose. So enzyme plus substrate, here is glucose, here is also sucrose. Same thing, enzyme plus product complex, you will find glucose and fructose. And thereby, if it is enzyme is present, that will give you a glucose and fructose. The free enzymes it will release as such. So this is the enzyme catalyzed reaction. This binding can induce a strain on the substrate molecules, destabilizing certain bonds and make them more susceptible to chemical reactions. Enzyme can also use various catalytic strategies to enzyme or to enhance a rate of reactions. For example, they can stabilize a transition state by providing a favorable microenvironment within their active site. As I said, this kidney light structure. So this is the mechanism by which enzyme catalyzed reaction is happening. Now I will give you how this reaction, this can involve the formation of bonding system in that. So as I said, from transition state and enzyme substrate complex, what are the involvement? So this can involve the formation of hydrogen bond. Another bond name, that is electrostatic interaction between that. Hydrophobic interaction is between the enzyme and substrate. So there are three things can happen. First, bonding with the hydrogen bond, bonding with the hydrophobic interactions, bonding with the electrostatic interaction with the hydrogen bonds, where in between the enzyme and substrate. Additionally, enzyme can use covalent catalysis also, where they form a transient covalent bond with the substrate during the reaction. Now, another important concept in enzyme catalysis is enzyme kinetics. These enzyme kinetics are very important to know every reactions with how the rate of reaction is changed, half of the maximum velocities, what is the involvement of enzyme substrate complex, how slope is shown between the substrate and enzyme. So generally, another important concept in enzyme catalysis is enzyme kinetics. You will find, as I said, factor affecting enzyme activity, pH, temperature, enzyme concentration, substrate concentration, inhibitors. Now I will also give you enzyme kinetics allows to understand how these changes in the substrate concentration and enzyme concentration. Two things. How the changes in the enzyme concentration and changes in the substrate concentration. Other factors also affect the rate of enzymatic reaction. The most commonly used most commonly mathematical models are used for enzyme kinetics are the Michaelis Menten equation and lineweaverburk plot. First is Michaelis Menten equation and lineweaverburk plot. These two, Burke plot for the enzyme inhibition and Michaelis Menten equations. So first of all, you have to understand what is V, what is V0, what is V max, what is Michaelis constant Km, and substrate you know S into a bracket. So for example, Michaelis Menten equation is equal to V0 equal to V max substrate divided by Km plus S. Now what is V0? That is the initial reaction, rate of reaction. Initial rate of reaction is V0. Remember. Then Vmax is the maximum reaction rate. What that is? That is the reaction rate where the enzyme is saturated with the substrate. If the enzyme and substrate is saturated, both are equal, then the Vmax, maximum rate of reaction. Vmax, please, substrate. Now K means Michaelis Menten constant or Michaelis constant, which is only determined by the nature of the enzyme. Remember these words. What is Km? Km is a Michaelis constant which is only determined by the nature of the enzyme and has no connection with the concentration of the enzyme. Remember these words. Has no connection with the concentration of enzyme, which is only determine the nature of the enzyme. It could be used to identify the different enzyme. Who? Michaelis constant Km is only determine the nature of enzyme. Therefore, it could be used as a identify the different enzyme. And as you know, so that can also be asked, write down the Michaelis Menten equation. So you have to write the initial rate of reaction V0 is equal to Vmx substrate divided by Km plus S. Now you have three figures with graph x-axis and y-axis, horizontal and vertical. So two line you can see with the color. One is red line, another is blue line, dotted with the red line, dotted with the blue lines. So slope change, y-intercept same. If it is interact all three, one up maximum, and V0 means initially rate of reaction, 1 upon V upon 1 upon S. If intercept with that, no inhibitor, rate is inhibitor presence and no inhibitors, then red blue light. If inhibitor, red line. Remember this, no inhibitor means blue line. Rate means y-intercept is change. So y-intercept, if it is change, then uncompetitive inhibitor. In that condition, you will find two things. Km means Michaelis Menten constant, which is only used to determine the nature of the enzyme, has no connection with the concentration of enzyme. So Km, Michaelis constant reduced, and half of maximum velocity. Weight Vmax means maximum rate of reaction is reduced in the condition of uncompetitive inhibition. And you have to write like this: dotted line parallel with the blue dotted line with the red dotted line. So you will like this: one strand here and here. Another, intercept all, slope change. There is y intercept no change. That is same slope or slope change, y intercept is same than the competitive inhibitors. So in that condition, you will find Km increased and Vmax unaffected. Rate of reaction, maximum reaction rate, or reaction rate, when enzyme substrate concentration is saturated with the substrate, is unaffected. Then enzyme and substrate concentration is saturated, it's reduced, then Vmax. Reduced means uncompetitive. Vmx reduced and Km unaffected, then it is called as non-competitive or mixed reactions, mixed inhibition. So that can also be asked in the examination. You can also read, as I said, instance notes in biochemistry. You will find the differences between these three, and you digitally or in written form they have written that. So you have to read like that also. But if it is asked in the examination, if they have not given a non-competitive inhibition, uncompetitive inhibition, or competitive inhibition, this condition, how to write lineweaverburk plot for the enzyme inhibition, you have to write all three. Then if only the figure is given, then you have to write where is the competitive inhibitor, where is the uncompetitive inhibitor, and non-competitive inhibitors. So three types is given. In every competitive inhibition, the two condition Km increased and Vmx unaffected. If Vmx is reduced, two things can be possible. But in uncompetitive inhibition, Km reduced. If Km is not unaffected, then you will tell as non-competitive inhibitions. Now in words, I will give you a Michaelis Menten equation. This relationship between initial rate of reactions V0 with the substrate concentration V0s, two kinetic constant Michaelis Km and the maximum rate reaction. So V0 is equal to Vmx S upon S plus Km, as I said in the previous slide. So Km value represents the substrate concentration at which the rate is. Now Km, you have to write like this: Km represent the substrate concentration at which the rate is half of max Vmax, and provide information about affinity of enzyme for its substrate. So repeat this word and you speak.
The Km value represents the substrate concentration at which the reaction rate is half of Vmax and provide information about the affinity of an enzyme for its substrate.
So in same reaction you will find Vmx. What is Vmx? So Vmx value, as I said previously in previous uncompetitive, non-competitive. So Vmx value represent the maximum rate. Remember Z, maximum rate at which an enzyme can catalyze a reaction when it is saturated with the substrate. Now, Lineweaverburk plot is a graphical representation of Michaelis Menten equation, as I said. and shown you it allows for easy determination of Km and Vmx value by plotting 1, 1 half or 1 by V0 against 1 by substrate. The slope of line is equal to Kmx by Vmax or y-intercept equal to 1 half of the maximum velocity. In recent years, the advances in structural biology techniques such as X-ray, crystallography, or you can say cryo-electron microscopy, okay, scanning electron microscopy, have provided detailed insight of three-dimensional structure of the enzymes and their complexes with the substrate and inhibition. This structural integrity or studies have a lot contribute to understanding these enzyme catalytic reactions and precise arrangement of amino acid residues within the active site and interaction that drive a catalysis. So, in overall conclusion of this enzyme catalysis is a complex, multifaceted field and encompasses various principles and concepts, as I said previously, lock and key models, induced fit models, and complementary explanation, as I said, enzyme substrate complex interaction with the different mechanisms that which facilitate the catalysis and including substrate binding, transition state stabilization, covalent catalysis, enzyme kinetics, allow enzyme kinetics that allows the quantitative analysis of enzymatic reactions that provide a valuable information about the enzyme substrate affinity and rate of reactions. So this is all about the enzyme catalysis